apologize again for class starting late. Can you hear and see me? You can't see me yet. Let's see here. Bear with me, I know you can't see me. And I have an idea why. See if I can get this fixed quickly. Dad?
Please leave your message for.
Sorry, bear with me just for a second because I know why you can't see me. Over Thanksgiving weekend, my dad was working on my computer and he did something and I can't remember what he did to make it so you can't view my camera.
Good morning.
Hi, is my dad available really quick?
I think he's talking to someone on another line. Hold on. Let me walk in there.
Okay.
Hold on. You on the phone? Mary?
Debbie? Dad, really
Hi.
quickly, what do I have to do to get my video on my laptop to show up again? What?
I think it was the function key. You have to look at the little function keys up there.
I tried that. It's not working, and I've got class right now. I'm in class.
Try it without the function. Are you holding down the function key?
I was.
Okay, that on your particular laptop.
Okay, he's
okay.
I'm trying, but I'm in class right now, and they cannot see me.
What's okay? It's an MSI.
It's an MSI. Yes.
Okay, he's looking.
I think when I let him play with my computer when we were at Melinda's house, he did something, and now it's not there at all. I still don't regret letting him play with my computer, but I don't know what he did.
Hold on, he's looking.
I got it.
Oh,
Debbie. I'm gonna hang up.
All right.
Love
Bye.
you. Bye-bye. All right. I'm sorry. I had to figure out what he did and what I needed to do to reverse it. I apologize. First, any questions or concerns? Can you hear me? See me?
Yes, I can hear you. I'm good.
All right. So if I remember right in the notes where we are, let me share my screen here. So where we are right now is we are working through the autonomic nervous system. We will finish that today, and we will start the last chapter, which is chapter 11. I noticed that not everyone has turned in the first case study. I think I received half of the case studies for the class. This is the first of 3 case studies, so the second one is already out there too. The reason you're given case studies is to apply your knowledge. What I want you to do, especially with the first case study, is use the PowerPoint. The questions were specifically selected from material that was given in the PowerPoint. The second one is a lot of more simply answered questions about the cranial nerves. If you can't find that information in between the lab and the lecture on the cranial nerves, which was chapter 14 and lab exercise 21, if you can't find it there, then Google it. But you should be able to find it there between those two sites. Those case studies altogether are worth about the full grade of one exam. If you look, they're worth 30 points each. Each one of your exams are worth 100 points. So altogether, all three of those case studies are worth 90 points. Since we are coming into the end of the semester, we have just a matter of about 2 or 3 weeks. It's coming down to where I'm trying to remember. I will be submitting grades on December 17th. If the material isn't done by then, I can't include it in your grade. So I need you to look at your grades, see what assignments you're missing, and let me know. What I need you to do is give me a list and tell me when you give me that list, when will you have it done by?
Hey, Professor, I have a question. I'm sorry. I just came into the video. About the case studies. The second one, is that the case study number 2?
Yep.
Okay, and when is that due? I'm sorry.
I don't, when did I have it due? I think I had it due like December 3rd. But
Okay.
it doesn't really have a due date. You'll see that I don't make those drop boxes so they close on a given date.
Right.
I just need you to get them done.
Okay. Okay, thank you.
I think I made it December 3rd. Your second one is going to be over muscle and the neuromuscular junction. And it's going to have to do with neurons and muscle. Assuming your third one,
Okay.
which I haven't posted yet.
So do we just answer the questions and then put it back in the Dropbox?
Yep.
Okay.
Yep. If, what was I going to say? Oh, I can't remember all of a sudden, but make sure that you're getting those in because altogether they are worth an exam grade. Approximately. Okay. Let's get started here if there's not any other questions. One other thing that you'll see here, and I did this because some of my classes are behind. I teach, this is one of 3 lectures that I teach for biology 121. And some of my other classes are behind with regards to the autonomic nervous system. So because I think it's one of the more easier chapters, and it's a relatively short chapter. It's the reason we're going to finish it today when we only started it the other day. I made a video recording of it. Of me just going through the PowerPoint. And they're right here and right here. This is the first part, which is about 30 minutes. This is the second part, which is about 60 minutes. If use them as a additional study tool for studying chapter 15 if you'd like. Let's see, where were we? It's 29, 28, 25. Here we are. So start at the very beginning. So something that we're going to learn right here is that the sympathetic, remember first, let me ask you, what are the two subdivisions of the autonomic nervous system, the two main ones? What are the two main subdivisions of the autonomic nervous system? Someone tell me.
Sympathetic and parasympathetic.
Correct, correct. The sympathetic and parasympathetic. The sympathetic is more a fight or flight. Parasympathetic is rest and digest. Parasympathetic is involved in developing your energy storages. The sympathetic is involved in expending those energy sources. With regards to anatomically how they're structured, if you look at going from the brain to the brainstem to the coccygeal region of the spinal cord, the sympathetic spinal nerves or the sympathetic nerves come off the spinal cord from T1 to L2. So from the thoracic nerve 1 to the lumbar nerve 2. So they're coming off the middle of the spinal cord. We often, because of where these sympathetic nerves come off the spinal cord, we often call this the thoracolumbar region or division of the autonomic nervous system. So they are distributed. Actually, I'm going to back up to one more slide here. The cell bodies are found within the lateral gray horns. You remember how we had the posterior and the anterior gray horns. And from L1 to T2, we also have lateral gray horns. In the lateral gray horns are where the preganglionic cell bodies are for the sympathetic nerves. That's also the reason you only see the lateral gray horn from T1 to L2, is because that's where the sympathetic nerves originate. And that's where they're located. And they're not found other places. So that's the reason we only see the lateral gray horn in that area. why do we need to distinguish? Do you remember why do we distinguish between preganglionic and postganglionic neurons in the autonomic nervous system? Why do we distinguish between those here? What does that mean? What does that mean? Someone tell me, why do we just, first off, what's a ganglia? Okay, in a minute, I'm going to put the name.
Collection of neurons.
Say it again. Say it one more time.
Collection of neurons.
It's a collection of cell bodies, and so it's a collection. You were close. It's a collection of cell bodies and dendrites and synapses. So where one neuron terminal, one axon terminal synapses with another cell body, and its dendrites. So if you have preganglionic neurons, those are the neurons that are before a nerve synapse. And postganglionic neurons are the neurons that are on the terminal end of that neuron synapse. So when we talked about the somatic motor nervous system, we talked about the fact that there's one neuron that goes out to an effector. However, with regards to the autonomic nervous system, there's two neurons. There's the preganglionic one and the postganglionic one. Because the pre is going to be like the presynaptic neuron. We've used that word before when we were, I think, in chapter 12. And the postsynaptic neuron. This time, because we know that synapses are going to occur in ganglia, the preganglionic neuron is the presynaptic neuron. The postsynaptic neuron is the postganglionic neuron. That's the way to think about this, is remember that there's two neurons, the preganglionic one and the postganglionic one. And the reason we call pre and post is because one's is the one going into is a presynaptic neuron. One of them is a postsynaptic neuron, and the synapse is occurring in a ganglion. Let me do one more thing here while we're at this. Oh, let me put this in words really quickly here. So two major distinctions, or a couple major distinctions. Somatic nervous system. Remember, this is your voluntary nervous system. Is your voluntary works on the skeletal muscles. So that's the target organ here. Works on the skeletal muscles. Voluntary works on the skeletal muscles. And there's one motor neuron. one motor neuron, remember, or one efferent neuron. It helps to think of it that way, one efferent neuron. So one neuron leaving the central nervous system and going out to the effector. One efferent neuron pathway. In the case of the autonomic nervous system, autonomic nervous system, it's involuntary. The target organs are cardiac and smooth muscle. And gland cells. And rather than there just be one outgoing neuron that goes out to an effector, which in the case of the somatic nervous system is the skeletal muscles, there's two neurons involved. Two neuron pathway. And if there's two neurons, there has to be a synapse between those two neurons, and that's going to occur in the ganglia. So when we think of a synapse, that's where the ganglia are. And so a presynaptic neuron is called the preganglionic neuron. And because ganglia are, because synapses are going to occur in ganglia. Synaptic neuron is going to be postganglionic. Are there any questions right now about the distinctions here? In the sympathetic nervous system, the preganglionic neurons are going to reach out to, the majority of them are going to reach out to the sympathetic chain ganglia, which is a structure that runs right alongside the vertebral column. And actually, I wish I'd pulled a picture of this. So give me 1 second here. Let's see here. If you remember the dorsal root ganglia, where that was, that it was right on that, adjacent to the spinal cord. And it was where the cell bodies of the sensory neurons were. This is similar. Ganglia. if you look here, here's the spinal cord. It's not a very big picture. I'm making it very big. But if you look right here, here's the spinal cord. Right next to it is the sympathetic chain ganglia. You see it right here too. So this is where the preganglionic neurons of the sympathetic nervous system are going to synapse with the postganglionic neurons. Within the sympathetic nervous system. Any questions? Because the sympathetic chain ganglia runs right adjacent to the spinal cord, and therefore the vertebral column, we also call it the paravertebral ganglia. It's right here. So the preganglionic neurons move into the sympathetic chain ganglia, and then they're distributed via the postganglionic neurons. Oftentimes, talk about where they're not, but many times from the sympathetic chain ganglia, as they pass through this, they're going to be distributed to every level of the body. So there's a high level of neural divergence from that point, or branching off. Because the sympathetic chain ganglia is right adjacent to the spinal cord, the preganglionic neurons are not very long. They're relatively short. They don't have to go very far. So they tend to be really short. However, the postganglionic fibers or axons are significantly longer. In contrast, when we talk about, I'm sorry, jumping forward when I shouldn't. The paravertebral ganglia, remember, so this is the same thing as the sympathetic chain ganglia. Paravertebral ganglia is the same thing as the sympathetic chain ganglia. Each paravertebral ganglia is connected to a spinal nerve via 2 branches. These branches are the branches of the rami communicante. If you go back to chapter 14, was it chapter 14? It was chapter 13. If you go back to chapter 13, we talked about the rami communicante there and how they were involved in the sympathetic nervous system. These are branches coming off the rootlets of the spinal nerves. So each paravertebral ganglia meaning each ganglion of this sympathetic chain ganglia are connected, connected to a spinal nerve by two branches, the branches of the rami communicante. The preganglionic fibers are small myelinated fibers. What do you remember about white matter versus gray matter in the spinal cord and in the central nervous system?
White has myelin.
Right. The white has myelin. That's correct, Rachel. The white has myelin. The gray does not have myelin. It is the same thing here. So the white communicating ramus is myelinated. The gray communicating ramus is unmyelinated. The preganglionic neuron is the white communicating rami. The gray communicating rami is unmyelinated, and it's the postganglionic fiber. Keep that in mind here. So the preganglionic neurons are the white communicating rami. The postganglionic fibers or neurons are the gray communicating rami. And the postganglionic fibers are going to extend the rest of the way to the target organs. That's the gray communicating rami. So the white rami communicante carry the preganglionic sympathetic fibers to the sympathetic chain ganglia. And the gray communicante carry the postganglionic sympathetic fibers from the ganglia to the spinal nerves. Is this clear?
Yes.
After entering the sympathetic chain ganglia, the preganglionic fibers can follow one of three courses. The first course is that they enter this sympathetic chain ganglia and immediately synapse with a postganglionic neuron. Immediately, the green here are the preganglionic neurons. The red is the postganglionic neurons. So what the first route, they can enter the ganglia and they synapse immediately. The target organs for this first route are sweat glands, pilorector muscles. Pilorector muscles are the smooth muscles in your that attach to the hairs on your arms and neck and so forth, give you goosebumps. They contract when you're cold. Sweat glands, pilorector muscles, blood vessels of the skin, and some skeletal muscles. That's the first route. The second route is that they can travel up and down the sympathetic chain ganglia and synapse at other levels. So remember this sympathetic chain ganglia, it's a chain and it's running from T1 to L2. So these preganglionic neurons can move up and down the chain and synapse at different levels. When this is occurring, their their target organs are going to be the iris, the salivary glands, the lungs, the heart, the thoracic blood vessels, and the esophagus. In the third route, they can pass through the chain completely and continue as splanchnic nerves that are going to lead to a second set of ganglia called collateral ganglia. And their target organs at that point become the liver, the spleen, the adrenal glands, the stomach, the intestines, the kidneys, the urinary bladder, and the reproductive organs. These collateral ganglia with this third route contribute to a network called the abdominal aortic plexus. If you remember, we talked about plexuses in the talked about plexuses with the brachial plexus, the cervical plexus, the lumbar plexus of the spinal cord. So this is a plexus of nerves. This collateral ganglia are contributing to a network of a plexus of nerves that are going to wrap around the abdominal aorta. Many of the postganglionic fibers are going to exit this plexus and accompany arteries of the same names and follow the same routes to their target organs. These are going to be involved, remember, in innervating the liver, the spleen, the adrenal glands, the stomach. So all essentially many things in the lower abdomen and pelvic region. So innervation to and from collateral ganglia. If the nerves pass through the thoracic ganglia, the 5 to 9 or 10, their collateral ganglia is going to be what's called the celiac ganglia, which is going to be in the postganglionic fibers are going to innervate the stomach, the spleen, the liver, the pancreas, the small intestines, and the kidneys. In the thoracic ganglia, 9 to 12, these neurons, preganglionic fibers, are going to synapse in the celiac and superior mesenteric ganglia and go out to the small intestines, the colon, and the kidneys. The lumbar ganglia, so L1 and L2, are going to synapse in the collateral ganglia of the inferior mesenteric ganglia. And the postganglionic targets are the rectum, the urinary bladder, and the reproductive organs. Any questions so far? So one exception to this two neuron pathway of the sympathetic nervous system is the sympathetic division's innervation of the adrenal medulla. Remember the adrenal gland, adrenal here. Renal means kidney, so adrenal is this gland on top of the kidney. The medulla, just like the medullary cavity of the bone, is the inner part of this gland. So the preganglionic sympathetic fibers that are going to innervate the adrenal medulla do not synapse within the sympathetic chain ganglia, just like those that are going out to the collateral ganglia. But they go directly to the adrenal medulla and they release acetylcholine. These preganglionic fibers are going to release acetylcholine within the adrenal medulla onto cells called chromaffin cells. These chromaffin cells are modified postganglionic neurons. They have to be modified neurons because if they can respond to a neurotransmitter, they have to be semi-light neurons. So they're modified postganglionic neurons. And these chromaffin cells are going to release epinephrine, what we often call adrenaline, and norepinephrine, which we call noradrenaline commonly. Questions about the different routes that these preganglionic fibers in the sympathetic nervous system can take? So there's essentially 4 different routes. Preganglionic fibers exit the spinal cord via the blank and pass through the blank to enter the what? And trunk here means chain. Let me call on somebody who I don't tend to hear a lot from lately. Quentin, what do you think the answer is?
C.
Why do you think it's C?
I'm not really sure. I'm just trying to take a guess. I'm just looking at the picture too, just to see if it will give me any answer at the right
Okay,
time.
this is the anterior surface. This is the posterior surface. What's the other word for anterior versus posterior? Ventral and dorsal. So is that, where is it coming off the door? Is it coming off the posterior side or the anterior? Looking here at this neuron.
Oh, hold on. Can you say that again?
Is it coming off the anterior surface or the posterior surface?
Anterior.
Okay, and is that, so that would be the ventral side. So it's going to be the ventral roots. You see why I'm saying that?
Yeah, I do know.
And the gray communicante ring, the gray communicante. Remember, they come off those gray and white communicante. So it's not the gray communicante, it's a white rami communicante. And then it's going to enter the sympathetic chain ganglia. Good try, Quentin. I appreciate it. So it's going to exit the ventral root, which is also called the anterior root. It's going to exit the rami communicante, pass through the rami communicante, and then enter the sympathetic chain ganglia or the sympathetic trunk ganglia. The primary reason that the sympathetic division of the autonomic nervous system produces immediate responses within the body is based on the concept of divergence. True or false?
True.
It is. It's true. It's true. Good job. It's true. Now, moving on to the parasympathetic division. So remember, we often call the sympathetic division the thoracolumbar division because it comes off from the thoracic region into the lumbar region. We often call the parasympathetic division the craniosacral because they're coming off the brain stem and brain and the sacral part of the spinal cord. Those preganglionic fibers are coming off this area, these two areas. The fibers of the parasympathetic division are going to travel through certain cranial and sacral nerves. In comparison to the sympathetic division, which has short preganglionic fibers, the parasympathetic division has long preganglionic fibers, whose axons are going to end in the terminal ganglia or near target organs. And they have short postganglionic fibers. So sympathetic chain ganglia sympathetic division preganglionic fibers, short preganglionic fibers, stop. Stop. My dog is licking me here, and I wanted her to stop licking me. Fibers and long postganglionic fibers. and the preganglionic fibers are going to exit via the rami communicante from the thoracolumbar region. Region of the spinal cord. Parasympathetic are going to have long preganglionic fibers Don't lick me. Stop. Don't lick me. Short postganglionic fibers. And the preganglionic fibers are going to come off the cranial, four of the cranial nerves. So cranial and the sacral region. So long preganglionic fibers and then short postganglionic fibers. And in contrast to the sympathetic division, there's very little neural divergence here. The four cranial nerves that are involved in the parasympathetic nervous system are the oculomotor nerve. Remember, that's cranial nerve number 3. That's involved in narrowing the pupil through its effects on the iris and focusing the lens. The facial nerve, cranial nerve number 7, is involved in innervating the tear, nasal, and salivary glands. The glossopharyngeal nerve is involved in innervating the parotid salivary gland, The vagus nerve is involved in innervating cranial nerve number 7. I can't believe I just said that. The vagus nerve is involved in innervating many of the viscera, and it's cranial nerve number 10. The sacral nerves that are going to be part of the parasympathetic nervous system arise from levels of S2 to S4, and they form the pelvic splanchnic nerves that are going to lead to an inferior hypogastric plexus of nerves. Hypogastric meaning part of the, underneath the stomach. These are going to be the nerves involved in innervating part of the lower intestinal region, and the bladder and the reproductive organs. questions? So the sympathetic nervous system originates off the thoracic lumbar region of the central nervous system. The parasympathetic nervous system is going to originate off the craniosacral region. The sympathetic nervous system, a lot of the major location of the ganglia is the sympathetic chain ganglia, in addition to some of the collateral ganglia, which we call prevertebral ganglia. The parasympathetic preganglionic fibers are going to have ganglia that are near or within the target organs, effector organs. Fiber lengths in the sympathetic nervous system, short preganglionic fibers and long postganglionic fibers. In the parasympathetic, long preganglionic and short postganglionic fibers. Neural divergence, as Quentin said, is extensive in the sympathetic nervous system and minimal in the parasympathetic nervous system. Because of the extensive neural divergence of the sympathetic nervous system, its effects are often widespread in general. Because of the minimal divergence in the parasympathetic nervous system, the effects are often more specific and local. One kind of division of the autonomic nervous system is the enteric nervous system. It's debatable whether we should consider this part of the autonomic nervous system or whether it's included or not. However, it is involuntary. That's the reason it's often included as being part of the autonomic nervous system. It's an involuntary regulated nervous system that's composed of 100 million neurons within the walls of the digestive tract. It is regulated by the parasympathetic and sympathetic nervous system divisions. The two plexuses that are part of the enteric nervous system are the myenteric plexus and the submucosal plexus. The myenteric plexus is involved in regulating gastrointestinal tract motility, while the submucosal plexus is involved in controlling secretions. Now, autonomic neurons, the sympathetic and parasympathetic have major contrasts in their functions. How can they innervate the same organs and have major contrasts in their functions and in what their responses, what's their actions? This is one of the reasons why this is possible, is because the postganglionic fibers of the sympathetic and parasympathetic nervous system are going to secrete different neurotransmitters. norepinephrine and acetylcholine. Also, the receptors on the target organs can vary significantly. Given target cells or effector cells can respond to the same neurotransmitter differently depending on what type of receptor they have for it. If you remember, neurotransmitters can have a wide variety of effects. Can have a wide variety of effects based on what receptor they bind to and what receptors are present to respond to that neurotransmitter. That's the major reason we see contrasting effects here, is because one neurotransmitter can be excitatory or inhibitory depending on the receptor that it binds to. Questions? Despite the variety of possible chemicals that could be used to transmit chemical messengers in the autonomic nervous system, only acetylcholine, norepinephrine, and epinephrine are used to any degree. There are well over 100,000 neurotransmitters in the human body, but only 3 are used to any degree here. The first neurotransmitter and the neurons that it secretes are the cholinergic neurons that are called cholinergic because they secrete acetylcholine. Synapses where acetylcholine is used are called cholinergic synapses. Acetylcholine can bind to either nicotinic or muscarinic receptors. All synapses for both the preganglionic neurons in both the sympathetic and autonomic divisions, and all postganglionic parasympathetic neurons are going to secrete acetylcholine. So all preganglionic neurons, all postganglionic parasympathetic neurons are going to secrete acetylcholine. Receptors that bind acetylcholine, the cholinergic receptors, have 2 types. We have muscarinic receptors, and these receptors are found on all cardiac muscle, smooth muscle, and gland cells. These, when acetylcholine binds to these receptors, it can either have an excitatory effect or an inhibitory effect due to the different subclasses of muscarinic receptors. Remember that these receptors are going to be found on the effectors of smooth muscle, cardiac muscle, and glands, and on sweat glands innervated by the sympathetic postganglionic axons. So all cardiac, smooth, and gland cells. Nicotinic receptors in are the other subclass. And they are always at the end of autonomic nervous system postganglionic neurons within the adrenal medulla. They are excitatory when acetylcholine binds. Always. When acetylcholine binds to nicotinic receptors, it's always excitatory. When it binds to muscarinic receptors, it can be excitatory or inhibitory. Nicotinic receptors. Do you remember why these are called nicotinic receptors? I know we talked about it when we talked about in chapter 12. Why are these called nicotinic receptors? These are receptors that combine nicotine. That's why they're called nicotinic receptors, is because nicotine mimics the effects of acetylcholine on these receptors. The muscarinic receptors are named for a muscarine toxin that combine to these receptors. Cause excitatory effects. So nicotinic receptors found at the end of all postganglionic neurons within the adrenal medulla. They're also found at neuromuscular junctions with skeletal muscle and are excitatory when acetylcholine binds. Adrenergic receptor synapses at which norepinephrine and epinephrine are used are termed to be adrenergic. If you think, remember the other name for epinephrine is adrenaline, so adrenergic receptors. These adrenergic receptors are within the adrenergic synapses for nearly all sympathetic postganglionic neurons. Any receptor that binds to epinephrine or norepinephrine is called an adrenergic receptor. We have two different divisions of adrenergic receptors. We have alpha adrenergic receptors. Alpha 1 are usually always excitatory. Alpha 2 adrenergic receptors are usually inhibitory. Beta adrenergic receptors, the beta 1 and beta 2 receptors can have different effects. They can both be excitatory or inhibitory. So in the sympathetic division, the preganglionic fibers are always cholinergic, meaning they're always secreting acetylcholine. The postganglionic fibers of the sympathetic nervous system are mostly secrete epinephrine and norepinephrine, but there's a few that secrete acetylcholine. In the parasympathetic nervous system, the preganglionic fibers are always secrete acetylcholine. And the postganglionic fibers always secrete acetylcholine. Questions? You guys are really quiet. Questions?
No.
One term that we need to bring in, define, and describe is what we call autonomic tone, which is the normal background rate of activity that represents the balance of the two systems according to the body's needs. So both nervous systems, both the sympathetic and the parasympathetic nervous system, always have some background rate of activity on the organs that they innervate. And if you remember at the very beginning of this chapter, we talked about dual innervation, because both divisions are often innervating simultaneously and activating simultaneously the same organs. And there's a constant balancing act between which nervous system subdivision is got the highest level of activity on a particular organ. So there's a balance between these two systems in their levels of innervation on given organs that changes based on the body's needs. So for example, the sympathetic and the parasympathetic nervous system both innervate the intestines. There's a balance there where, depending on what is going on in the environment, if you're running away from a bear or not running away from a bear, the sympathetic division will have a higher level of innervation at that moment when the bear is there. And when the bear is not there anymore, then the parasympathetic nervous system will have the higher level of activity. So there's a balancing act here. The sympathetic nervous system keeps most blood vessels partially constricted and works to maintain blood pressure. The parasympathetic division maintains smooth muscle tone in the intestines and holds the resting heart rate down. An example of this is with regards to heart rate. Both the sympathetic and the parasympathetic nervous system are involved in regulating the heart rate. The heart has, and one of the very first chapters of that is covered in 122 is talking about the heart. And the heart has its own autorhythmicity, so it sets its own heartbeat. It regulates its own heartbeat. It has the ability to maintain a heartbeat without any central nervous system input. So it has an intrinsic heartbeat to it. The parasympathetic acts on it too, and the sympathetic simultaneously acts on the heart. The parasympathetic works to lower that inherent heart rate. And the sympathetic nervous system works to increase that heart rate. Questions? We're almost finished with this chapter, and then we'll take a 10-minute break. But I want to finish this chapter first. Influences on the autonomic nervous system. The autonomic nervous system is regulated by feedback loops between the spinal cord and brainstem. However, these feedback loops between the brainstem and the spinal cord also receive input from the hypothalamus. The hypothalamus is the main regulator of these feedback loops. And it receives input. Hypothalamus receives input from the limbic system and the cerebral cortex. If you remember when we talked about the hypothalamus in the chapter 14, we talked about the fact that it's a major integrator of the autonomic nervous system because it's the boss of these feedback loops between the brainstem and the spinal cord. And it receives some input and from the cerebral cortex and the limbic system. Remember, the limbic system is your emotional learning center. So there's input coming from the cerebral cortex and the limbic system, which are affecting the hypothalamus, which is the integrator and the regulator of the feedback loops between the brainstem and the spinal cord. Questions? So we've talked a lot about dual innervation and the fact that many organs are simultaneously innervated. And, you know, that autonomic tone and so forth. There's at least one exception of where the sympathetic nervous system is the only innervator. And that is that in the blood vessels. The sympathetic nervous system is the only innervator of regulating blood vessel diameter. And so when the sympathetic nervous system is firing levels to the blood vessels increases, vasoconstriction occurs. When its firing levels decrease, vasodilation occurs. So the diameter of the blood vessels is only proportional to the level of sympathetic nervous system firing. Because there is no parasympathetic innervation here. many of our drugs that we take and that are developed target our own human cells, and many of these are acting on neurotransmitters. They can either enhance the effects of a given neurotransmitter, or they can inhibit the effects of a neurotransmitter. So typically when a neurotransmitter binds to a receptor, it binds to the receptor and then the receptor produces an effect. It's followed by a response. That binding produces a response. When a drug is an agonist, a drug agonist drug, can bind to the receptor and activate that receptor to have the same effect it would if the neurotransmitter bound. This is what we call a drug agonist. It binds to that receptor and produces the same effect as the neurotransmitter normally would. We also have drug antagonist drugs that are similar in shape enough that they bind to that receptor. However, when they bind to that receptor, the receptor does not have the effect it would. When it's not activated by the binding of that drug. It's just that at that point, the active site is blocked to anything else binding to it. And so there's no effects produced when antagonist drugs bind. It's like an antagonistic inhibitor. It doesn't have any effect. So the only thing that an antagonist drug does is block that receptor from binding to its natural neurotransmitter. We have other drugs out there that that are antibiotics that are targeted against pathogenic cells. But many of our drugs work on our own human cells. In the context of these drugs, one of the ways that neurotransmitters, let me put it differently. If you remember from chapter 12, when we talked about neurotransmitters, we want them around, but then we want to be able to get rid of them too, because we don't want them always firing and always causing our neurons to be always active. We need the neurotransmitter to be around and then go away. And if you remember, there were 3 ways that it could go away. The neurotransmitter could either be retaken up into the presynaptic neuron, it could be broken down, or it can diffuse out of the cleft. One of the ways that neurotransmitters are broken down, monoamine neurotransmitters are broken down, is with this enzyme monoamine oxidase. So some monoamine neurotransmitters are norepinephrine, epinephrine, dopamine, serotonin, and histamine. We have drugs that specifically inhibit this enzyme and slow down the breakdown of these monoamines. And by slowing down the breakdown of these neurotransmitters, monoamines, the neurotransmitters are left in the synaptic cleft for longer. because they don't get broken down as quickly, the neurotransmitters. A few examples. Prozac, the generic is fluoxetine, is an inhibitor of this monoamine oxidase that breaks down serotonin, therefore keeping serotonin around in the synaptic cleft longer. Levodopa works to block monoamine oxidases from breaking down dopamine, keeping dopamine in that synaptic cleft for a longer period, and is used to treat Parkinson's disease. Questions?
No.
Why are amphetamines so easily addictive? If you've heard the reason that they regulate allergy meds is because pseudoephedrine can be made into methamphetamines.
That's what they put.
Say it again.
That's what they put in nicotine.
No, this is in allergy meds. If you're like me and you take an allergy med every day, when you get your prescription or you get certain allergy meds, you have to show your ID because it's regulated. And if you have over,
Sorry, I meant alcohol.
it's okay. If it's over a certain quantity for the month, unless you have a prescription for it, you can't get it. Because of the abilities to make methamphetamines from pseudoephedrine, which is in many decongestion allergy meds.
So are allergy meds, like, can they be addictive then?
No,
I've never heard of anybody being addicted to allergy meds.
they're not addictive, but because you can convert the chemical molecule of pseudoephedrine into meth, that's the reason they regulate it, because people will buy the allergy med to make it into meth.
Ooh.
Yeah, if you've ever, that's the reason they just within the last couple of years they started regulating allergy meds.
That is crazy. All right, I never knew that.
So why is it so addictive? For this reason. These methamphetamines chemically resemble the monoamines associated with elevated moods. You're a lot happier, and we're energized. They work because they chemically resemble these neurotransmitters. They can inhibit monoamine reuptake, promote monoamine release, and inhibit this enzyme. So inhibit the breakdown, keeping it around even longer. That's the reason that methamphetamines can be so addictive. Beta blockers are, remember, the adrenergic receptors that are beta enter the bloodstream via the gastrointestinal tract. And they prevent norepinephrine from binding to beta adrenergic receptors on the heart muscle and in the walls of blood vessels. Normally, norepinephrine on the heart is excitatory. But when these blockers are there, they block those receptors. So even though the norepinephrine is being released on the heart muscle, the heart doesn't respond to it because the receptors are blocked. So even if the heart gets activated, but even though the neurons are firing on the heart to activate the heart rate stays normal because the receptor is being blocked. So the result is that there's a decreased heart rate and there's decreased cardiac output, which is the amount of blood pumped per minute from the heart. And last slide here. Something that most of us struggle with, and many of us, if not most of us, struggle with an addiction to caffeine. Whether you realize it or not, it is an addictive substance. I don't drink coffee. I think I've mentioned that before, but I don't drink coffee. But I can tell you that I wake up with an energy drink in my hands. And I use it to wake me up and jolt me up because of the caffeine. I wouldn't drink them if they didn't have caffeine in them. Why can we struggle with this dependence on caffeine? Caffeine mimics an excitatory neuromodulator called adenosine. When it binds to the receptors for adenosine on the neurons, when caffeine binds to these receptors, actually, let me say this differently. When caffeine is actually a drug antagonist against adenosine, because when adenosine binds to the receptors for acetylcholine, it causes increased acetylcholine secretion, resulting in vasoconstriction. What caffeine does is it blocks those receptors for adenosine. And so even though adenosine is present, the vasoconstriction is not going to occur, and instead vasodilation is going to occur. And you're going to get increased blood flow, which is essentially mimicking one of the effects of the sympathetic nervous system, vasodilation. So it lowers that neuron's ability to initiate action potentials, thus decreasing the acetylcholine secretion, thus maintaining more vasodilation. Get increased vasodilation, which makes you feel more alert. Questions?
No.
Okay, you guys, let's take 10 minutes and we'll come back and start chapter 11. Did I not download chapter 11 here? I don't see it. Here, let's see if we can do something quick. Somebody's got the snow, but I didn't see it. I'll snow. Aaron. Yeah. Other, not yuck. You can pawn them. Not yuck. I realized when I went to open chapter 11 here that it wasn't posted. So I just posted it. Let me see. It's here yet. 1 second here. Flash. Just posted it. So let's see. Why is it not there? I just posted it, didn't I? Let's look. Wait 1 second here, since I know I just posted it. There it is. Move to, I don't know why it got moved there, but we're gonna move it here. So muscular tissue, chapter 11. Overview of muscle tissue. Body movement results from alternating contraction and relaxation of muscles. Skeletal system provides a leverage and a supportive framework for this movement. Myology is the study of muscles. So your muscles use your bones as levers to provide movement. Whether it's locomotion, chewing, snapping your fingers, your muscles are using your bones as levers. There's three basic types of muscle. Skeletal muscle, which is found throughout the body that's attached to the skeleton or the bones. Cardiac muscle that's only found in the heart. And smooth muscle that's found within the walls of the thoracic and abdominal viscera, meaning the organs, and the surrounding blood vessels. Skeletal muscle is attached to the skeleton, and its main functions are motion, so walking, running, chewing, snapping your fingers, maintaining posture, keeping you upright, producing heat, and thermoregulation, so maintaining temperature. In appearance, it's got multiple peripherally located nuclei. And it's striated. It's got alternating light and dark bands that can be observed underneath the microscope. It's voluntarily controlled. And there's some subconscious control. If you think about your somatic reflexes, the patellar reflex, unconsciously controlled. Your diaphragm, which is your major muscle of breathing, is is a skeletal muscle. The muscles for posture, you don't have to consciously think about contracting and relaxing them. So they're also involuntarily controlled. Cardiac muscle, on the other hand, is within the walls of the heart. It pumps blood. It has one centrally located nuclei. It's also striated. And has intercalating discs, which are gap junctions. Remember what gap junctions are? They're little tunnels between cells. Gap junctions that join neighboring fibers and allow for direct communication between fibers. Ways to distinguish. The major way that I can distinguish when I'm looking at a slide of one muscle, either cardiac or skeletal muscle, is the intercalated discs, because you don't have those in skeletal muscle. They allow for the heart, all of the heart muscle to contract at one time and relax at one time, because of the communication that goes on within those intercalated discs, those specialized gap junctions. The cardiac muscle is under involuntary control because the heart displays that autorhythmicity. Talked about this a little bit in the last chapter. You don't have to think about your heart contracting and relaxing for it to contract and relax. It can speed up or slow down when influenced by neurotransmitters, remember the autonomic nervous system, and by hormones. A smooth muscle, in contrast, is found within the walls of the blood vessels and internal hollow organs and structures, for example, the GI tract, and within skin where it's attached to hair follicles. It has a characteristic ability to perform peristalsis, which is moving things along a tube of muscle. It's a rhythmic contraction and relaxation of muscle along a tube, like the GI tract, gastrointestinal tract, to move substances down the tube. Leo's toe. Sorry, watch my cats fight right next to him. In appearance, there's no striations. And there's one centrally located nucleus. And it's under involuntary control. It's regulated by the autonomic nervous system and hormones. Here's some different distinguishing characteristics from the major, comparing and contrasting the different muscle types. The majority of this chapter is going to focus on skeletal muscle. Talk a little bit about smooth muscle, a little bit about cardiac, but the majority is going to focus on skeletal muscle. Remember, these are the target organs or effector organs of the somatic nervous system, is skeletal muscle. Each skeletal muscle fiber arises during embryonic development from the fusion of 100 or more myoblasts. Myo means muscle. Every time you hear the word myo, Myo means muscle. Do you guys remember what does blast mean when you hear it on the end of a word? You remember we saw it when we talked about osteoblasts. What are myoblasts? Say it again, Aaron.
It creates the cells?
It creates, it's a building. So myoblasts are going to be progenitor cells to muscle cells. Building. One other word that I'm going to introduce here is sarco. So sarco also means muscle. So myo and, actually, let's put it up here. Myo and sarco are both going to refer to something as being part of muscle. So each skeletal muscle is going to arise from the fusion of 100 or more myoblasts. So because each of these cells has a nuclei, the mature muscle fiber that's formed from the fusion of all these myoblasts can have more than 100 nuclei. Satellite cells are unspecialized myoblasts that can be used to regenerate damaged muscle fibers. This is why muscle can have abilities to regenerate, is because of the satellite cells. Cell division, once the fusion of the myoblasts occurs, cell division ceases. The number, the number of muscle fibers that you have when you're born is all you will ever have. Most of these cells will last your lifetime. So muscle cells do not actively divide. Keep that in the back of your mind. If you think about muscle enlargement, we can get bigger muscles. Can get bigger muscles, but it's not because the cells are dividing. Muscle growth is occurring because of hypertrophy, meaning the cell itself is getting bigger. The cell, it is enlarging because of an increased number of myofibrils within the muscles fibers. So these are little segments within the cell that can increase in number. So the hypertrophy occurs because of the increased numbers of these myofilaments or myofibrils that are made of myofilaments. Testosterone and human growth hormones stimulate hypertrophy. Some different properties of muscle tissue. Muscle tissue is excitable. It is responsive to chemical signals, stretch, and electrical changes that occur across the membrane. It's conductive. Local, just like your neurons. Local electrical change triggers a wave of excitation that travels along the muscle fiber. Muscle cells can generate and propagate action potentials. Muscles have contractibility, which means that they have the ability to shorten when stimulated. There's two different types of muscle contraction. There's what we call an isometric. Iso meaning to stay the same. Metric meaning length. So stay the same length muscle contraction. Muscle develops the tension, but it doesn't shorten. Some examples of this would be your posture muscles, muscles of your back. They have tension on them, but they don't shorten. Isotonic muscle contraction is tonic having to do with the tension. So there's enough tension generated by the muscle to move an object. So then the muscle shortens. The tension remains the same and the muscle shortens. Other properties of muscle, it's contractile, it has the ability to shorten. It also has the ability to be stretched between contractions without being damaged. One of the major genetic diseases is when the muscle can't stretch without being damaged. We'll see this later in the chapter. Muscular dystrophy. Elasticity. It can return to its original resting length after being stretched. Which of the following is not a function of skeletal muscle tissue? Moving your eyes from left to right as you read this question. Moving your head up, shivering to generate body heat when you've become chilled, moving food or stabilizing joints to help maintain body positions, which is not a function of skeletal muscle.
The intestines one,
Why do you say that, Erin?
because while there is muscle there that helps to move things, it's not skeletal muscle. There's no leverage and things involved.
You're right. Let me give you another reason for why you're right. What type of muscle is found in the gastrointestinal tract, which
Smooth
is the
muscle.
small intestine? Smooth muscle. So it's not skeletal muscle, like Erin said. Muscle is organized in what are called fascia. Fascia wraps around the muscles and other organs of the body. So this is like a sheet of irregular, dense connective tissue that is that's deep within this, deep to the skin, and it surrounds muscles and other organs. There's also a subcutaneous layer of fat that separates the muscle from the skin and protects the muscles from trauma. So if you look here, here's the subcutaneous layer. And the fascia are surrounding bundles of muscle fibers. This fascia lines the body wall and limbs, holds the muscles together with similar functions, allows for free movement of muscles. You can move. I'm a scrawny person, but you can move. Here's my biceps brachii right here. I can move it a little bit. You can move it a little bit. Allows for free movement of muscles. This fascia also carries nerves, blood vessels, and lymph vessels, and fills the spaces between muscles. These fascia or fascicles give muscle its characteristic grain. Remember that sarco was one of the words that refers to muscle. What's lemma? Do you remember that keyword from before, that base word? Or root word? Lemma means a plasma membrane. So a sarcolemma is the plasma membrane of a muscle fiber. Muscle fiber is synonymous. Muscle fiber is synonymous with muscle cell. So the sarcolemma is the plasma membrane of a muscle fiber. The sarcoplasm is the cytoplasm of the muscle fiber. Remember plasm, cytoplasm, sarcoplasm. The myofibrils are long protein cords. Remember, these are the ones that are going to increase in number if the muscle cell hypertrophies. So the myofibrils are long protein cords that occupy most of the sarcoplasm. The glycogen is a carbohydrate for ATP synthesis. It's a stored, remember, glycogen is a stored glucose molecule. So it's a carbohydrate that's going to be used to produce ATP, which is a requirement for muscle contraction. Major requirement. Myoglobin. You've heard the word hemoglobin? Maybe. Myoglobin is hemoglobin derivative or hemoglobin member that binds to oxygen and releases it within the muscle for ATP production. It's the red pigment. It's what gives muscles their red color. You've seen white meat and dark meat. We all had Thanksgiving a week ago. Hopefully you got some turkey in there. Dark meat has more myoglobin in it. white meat has less. Think about that throughout this chapter. Why does white meat have less myoglobin in it? Why does dark meat have more? The mitochondria lies in rows along the muscle fibers and is located close to the contractile muscle proteins that use ATP. Mitochondria, remember, that's the powerhouse for producing ATP. So it lies close to the contractile proteins that are directly involved with contraction. So the ATP can be produced as quickly as needed. The sarcoplasmic reticulum is a modified smooth endoplasmic reticulum that forms a network around each myofibril. The terminal cisternae, right here, terminal cisternae are dilated end sacs of the sarcoplasmic reticulum, which cross the muscle fiber from one side to the other. These sarcoplasmic reticulum act as a calcium reservoir and release calcium through channels to activate contraction. We'll talk about how that works between on Thursday and next Tuesday. Transverse tubules or T-tubules are tubular enfoldings of the sarcolemma plasma membrane, where the tunnels form into the plasma membrane through which action potentials travel and spread throughout the muscle fiber. This is a triad, meaning T-tubules with terminal cisternae associated with it. So remember what the terminal cisternae are. They're the dilated end sacs. And the transverse tubules are the tubular enfoldings. So here's the terminal cisternae, here's the T-tubule. And all three of these are called a triad. Allowing the action potentials to travel and spread throughout the muscle fiber. Myofibrils are contractile organelles of skeletal muscle. They contain the thin and thick myofilaments arranged in units of sarcomeres. Remember, this is what enlarges. These are the myofibrils are the ones that increase in number when muscles get bigger. Born with the same amount of muscle cells, they just enlarge. The reason they can enlarge is because we can increase and decrease the number of myofibrils. These contractile organelles of skeletal muscle contain thick and thin myofilaments arranged in units called sarcomeres. Right here to right here is one sarcomere. Right here to one here is a sarcomere. The striations make the muscle appear striped. You look here, here's one stripe, here's the next stripe, here's the next stripe. That's why it looks striated. Sarcomeres are those compartments of arranged filaments that are going to extend from one Z disc to another. And Z discs are the boundaries of the sarcomeres. Looking right here, here's one Z disc. Here's one Z disc, here's the next Z disc. Here's one Z disc, here's the next Z disc. And the myofilaments are arranged in here. And from one Z disc to another Z disc is one sarcomere. Which structure that we've talked about within this chapter releases calcium to trigger muscle contraction? Somebody tell me. Go back and look at the slides that we've just gone over and tell me which structure is going to release calcium.
The sarcoplasmic reticulum.
Yes, exactly. The sarcoplasmic reticulum. Why do you think calcium would have to be actively transported back in? Guys, do you remember when we talked about active transport versus passive transport? I know it was a long time ago. This was like chapter 3. Active transport versus passive transport. What does active transport mean?
Active transport requires ATP or some type of energy to move the molecules across the cell wall.
And why does it require ATP?
Because it's either moving it like against its concentration gradient or other things like that.
Good job, exactly. So it's moving it against its concentration gradient. As we go through this chapter, answer, why does calcium have to be moved across against its concentration gradient? So 3 types of muscle proteins are going to comprise the myofibrils. We have contractile proteins, which cause the muscle to shorten when they interact with one another. Myosin and actin. We have regulatory proteins that regulate the interactions between the contractile proteins, tropomyosin and troponin. And we have structural proteins that keep filaments in proper alignment, gives the myofibrils their elasticity, and links the myofibrils to the sarcolemma. The structural proteins include titin, alpha-actinin, myomesin, nebulin, and dystrophin. These are structural proteins, regulatory proteins, and the contractile proteins. So myofibrils are bundles of myofilaments, which are myosin and actin, that fill up most of the sarcoplasm, the cytoplasm within the muscle fiber. They are surrounded by sarcoplasmic reticulum and mitochondria. Their striated appearance is due to the orderly overlap of the thin and thick filaments. Myosin is the thick filament, actin is the thin filament. So myofilaments include myosin, is the thick filament, and actin is a thin filament. One word that we'll see, that we'll talk about, two other distinctions that we'll talk about here in a second. Myosin represents the majority of the A band. And actin represents the majority of the I band. So their striated appearance is because of the orderly overlap of the thin and thick filaments. The thin filament meaning the actin, the thick filament is the myosin. Thin filaments, thick filaments, and they're overlapping. The whole reason that muscle contracts is because of this overlap. The thick filaments are made of several hundred myosin molecules. Each molecule is shaped like a golf club. Two chains of myosin are intertwined. Two myosin filaments are intertwined to form a shaft like a tail. And then there's a double globular head. Double globular head. The heads are directed outward in a helical array around the bundle. These myosin molecules function as a motor protein, converting ATP to motion, converting the potential energy of ATP to kinetic motion. The thin filaments are actin, which are two intertwined strands of, and each strand is made of globular actin subunits with an each subunit having an active site that can bind to the head of the myosin molecule. In relaxed muscle, this site is blocked by tropomyosin. Remember, tropomyosin and troponin were regulatory proteins. Right now, do you have any questions for me? Let's go for a few more minutes here, just 4 more minutes. The troponin and tropomyosin act like a switch that determine when the fiber can or cannot contract. It works like a light switch. Contraction is activated by the release of calcium into the sarcoplasm and its binding to troponin. Troponin changes shape and moves the tropomyosin off the active sites on actin. So remember that tropomyosin hides the active sites on actin. When troponin binds to calcium, it shifts, removing the tropomyosin from off these active sites. A structural protein involved here is titin. Titin is a huge, springy, the light green here, huge, springy protein that runs through the core of the thick filaments and anchors the thick filaments to the Z disc and the M line, and helps stabilize the position of the thick filaments. Its ability to stretch is what gives muscle its extensibility. The M line is the middle of the thick band. Think of M as being the middle. So titin is what provides recoil to give muscle its elasticity. So it stretches, giving muscle its extensibility, and it provides recoil to give muscles their elasticity. The Z disc is made up mostly of a protein called alpha-actinin. Remember, that's another structural protein. So here's the titin, here's the Z line, Z disc. This to this is one sarcomere. Here's the thin filaments, here's the thick filaments. The M line is made up of accessory proteins like myomycin, C proteins, and creatinine kinase, which is a functional enzyme that are also connected to titin. You have any questions for me? I will see you on Thursday. Have a wonderful 2 days. You're welcome.
Thank you.
See you then.
See you then.
I just have one
Go
question.
for it.
So for our exam that's due tonight, do you know how many questions that is?
I think it's 82, like the other
Okay.
one was.
Okay. That's all. Thank you.
Genesis, have a good day, okay?
You too.